Good evening. My name is John Stroh. I am with Nanospectra Biosciences. I was just thinking as we were going through the presentation that when you're the last speaker, there's always a little bit of difficulty in, first of all, keeping the attention of the crowd. Everybody's probably getting a little hungry about now. The 5 o'clock espresso caffeine is probably worn off. And to top that off, just before I came up, my COO, Kevin McNerney, who's here, asked if I could speed it up because he was missing the Jersey Shore reunion. So my presentation, first of all, I'm not a professor at UCI. In fact, I don't have a PhD. You will not see any Racka Welch pictures, any cute kittens, references to Sholenkov, if I pronounce that right. Sholenkov. And to top it off, I'm going to be talking about a commercialized company I'm CEO of, that the technology is a spin-off from a university in Texas, of all places, Rice University. So I'm kind of contra of everything that's been discussed so far. But what I am is a CEO of a company, an early stage company. I've worked primarily with early stage companies, both on the operations side and on the investment banking side. I've raised over $1 billion for healthcare companies, primarily early stage life science and device companies. And I've been part of 5 successful exits. So what I'm going to focus a little bit about is our technology specifically, and a little bit about the commercialization process. What happens after all these great ideas that are developed at places like UCI get into the commercial application? How do you make them successful? How do you get to funding? How do you get them to approval and ultimately commercial success? So Nanospectra Bioscience is very simply, it's a cancer therapy using precise thermal destruction of solid tumors through nanotechnology by the use of infusion of gold nanoshells. These were developed in the lab of Jennifer West and Naomi Hallis at Rice University, 2 of the early pioneers in this technology. Interestingly enough, although it is infused into the patient, it is considered a medical device. So the particles are inert, and we activate those through the use of a laser. Why that's particularly important is it's a heck of a lot easier getting approval for a device than for a therapeutic. Nothing's easy these days through the FDA, but it is easier with medical devices. Our therapy has broad applications. It's a platform technology. Can be used in all solid tumors in cancer. Currently, we are in the clinic with head and neck cancer, prostate cancer. We have done a multitude of animal work in glioma, pancreatic cancer, breast cancer, and we will shortly be going into lung and melanoma. So, very excited about where we are. And recently had some, actually yesterday had some very good data from our trial, our prostate trial. So, which was, if we hadn't gotten that good data, then we'd have to rethink our prostate application. So that was particularly good news. The clinical advantages of our technology, there's a few, and really precision, the very focalized application of the therapy. So as I describe it in more detail, you'll see that specifically in treating cancerous tumors that are irregularly shaped, are non-resectable or inoperable, and in cancers such as prostate cancer, which is very focalized, it's a way to specifically ablate the tumor without impacting the healthy tissue. So again, with prostate, you're able to kill the cancerous tumors without impacting the nerve bundle, which is the primary source of the pretty significant side effects in prostate cancer treatment. And there are other adjunct therapies that our technology is applicable to. For instance, our technology enhances radiation treatment and enhances chemotherapy treatment as well. Very simple, our oral laser therapy system. We have the nanoparticles that are infused into the patient with a cancerous tumor. We use an interstitial fiber optic probe, 8 nanometer laser, FDA cleared. So again, really off-the-shelf technology in conjunction with our Orishell or nanoparticle therapy. Again, you're talking very, very small nanoparticles, 150 nanometers across, 50 nanometers in the center. So one of the primary questions is, okay, there's a lot of ablation technologies. What's special about what we're doing, and how does it fit the unmet need? Well, again, as I mentioned, very tumor specific, minimizes the collateral damage, and effectively heats and kills the tumor without raising the temperature, or very minimally for a very short period of time, raises the temperature around the tumor. So very minimal, if any, heating to healthy tissue. So I'll give you an example. Here's a tumor that looks more like someone hitchhiking, but here's a tumor with irregularly shaped margins. Typically, a surgeon will, once he excises the tumor, very frequently there is still residual disease when you have irregularly shaped tumors. Other ablation technologies, there's ablation zones. So as the physician ablates these various zones, you still may have residual disease as a result of the irregular shaped tumors. So in our technology, essentially what we're doing is we're infusing the nanoshells anywhere from 12 to 24 hours prior to surgery. And because of the leaky vascularity of a cancerous tumor, the very fast growth, the nanoshells are attracted to the cancerous tumor. So as you can see, as the nanoshells are released into the bloodstream, once they hit that leaky vascular tumor, they will accumulate within the cancerous tumor. So we use a near-infrared laser to activate the nanoshells, ablate the tumor, and in effect what we have done is define the margins more specifically and lessen the potential of reoccurrence. Here's an example of a study that we've done with dog brain tumors. You can see that the tumor site itself, it's particle-focused ablation, so that temperature is risen to the point of ablation, where the healthy tissue has increased less than 10 degrees for a very short period of time, so no damage occurs in the healthy tissue. Here's an example of a study we did with mice. And if you look at that top line, the green line, you can see the Orishell treated mice lived beyond the 56 days. We had one that died, but unrelated to the therapy itself. And in comparison with sham treatment, untreated control, the effects and the efficacy of the Orishell treated patient was quite significant. Excuse me, patients, mice. So again, I mentioned adjunct to current therapies. I won't go into too much detail. This is in collaboration with research done at MD Anderson, but effectively when a tumor is removed and the area is radiated, there are still some vascularity within that site that is virtually hidden, that has the potential to reemerge and reoccur. So by actually warming the area as opposed to heating it to an ablation temperature, you're able to kill those cells and in effect increase the efficacy quite significantly, both in radiation and in chemotherapy. We've done a series of collaborations primarily within Texas, but I think one of the interesting things about Houston is the fact there's a lot of institutional collaborations. A lot of it's because of the proximity of Baylor, MD Anderson, Rice, etc. But one of the things that we want to do as we move forward is work with institutions like UCI, continuing collaboration. There's a lot of great work being done here, and we'd like to be a part of that, and as a way to complement our therapy with therapies that are being done here. We also are working with some commercial companies. We've had interest from commercial companies. Sebacia is one that's actually using nanoshells to basically turn off the sebaceous gland for acne. So basically, essentially does what Accutane does without the side effects. Cancer portfolio, again, won't have to go through these numbers, but it's a huge market and a significant commercial opportunity for us. Intellectual property, we're using Kenobi as our IP attorney, but very, very strong licenses, most of them from Rice, UT Austin, and we're going to continue to broaden that application. As the comment was made before, the IP portfolio of a young company is their primary resource, so we certainly recognize and will continue to strengthen our portfolio as we go forward. Won't go into details, but we have a strong management and board, and we'll continue to look for additional resources as we grow. We've raised about $8 million to date. A lot of our funding has come from grants. So just as a conclusion, when you look at nanomedicine, the opportunity, these particle-based therapies have enormous potential, and in a lot of areas where traditional medicine has failed in therapeutics. Venture investors funded over $800 million in 2010. 54% of that went to nanomedicine companies. Many clinicians see nanotechnology as really the ideal solution for the detection and treatment of many diseases. So as more nanotechnology companies emerge from successful clinical trials, the level of venture investing will continue to accelerate. So the early funding through friends, family, individuals, grants, angels, the traditional, as we were talking earlier today, it's unfortunate, but the venture and venture capital seems to have been eluded somewhere along the way. But there is money out there. There is opportunities. We are successfully moving down the path, and my, I guess, message to leave with those who are looking to commercialize opportunities are: be creative, look at new funds, smaller funds. It's still an emerging technology, although it's been around for quite a while, and I think you'll see significant commercialization opportunity, or significant commercialization results in the coming years. So we're excited and look forward to hearing more and exciting things coming out of not only our company, UCI, but nanotechnology, nanomedicine in general. So thank you very much.